Where else might life exist in our solar system? That's a question that NASA scientists are trying to answer. An intriguing place we are studying is Saturn's largest moon, Titan. It's the only body in the solar system, other than Earth, to feature seas of liquid on its surface. But that liquid is methane instead of water. The cell membranes we have on Earth, part of our building blocks for life, could not form here. Previous computer simulations investigated which chemicals could be used to make cell-like membranes on Titan. It's been calculated that this molecule, acrylonitrile, is the best candidate to form the most stable membranes. What's exciting is that our team at NASA Goddard has now detected large amounts of this chemical in Titan's atmosphere using spectroscopic data from ALMA, the Atacama Large Millimeter Submillimeter Array, located in Chile. And since Titan has rain, a significant amount may be reaching the surface and ending up in Titan's vast lakes. More research is needed, but by definitively detecting this astrobiologically relevant molecule on Titan, we are another step closer to understanding how and where life could exist in our solar system.
Well, hello, everybody. This is Scott Roberts from Explore Scientific and the Explore Alliance, and it's my pleasure to introduce Dr. Caitlin Ahrens, who will be introducing her special guest. If you haven't already guessed it, the topics will be about, I'm sure, about astrobiology and maybe the ocean worlds, maybe with a little bit of focus on Titan. It is, these other worlds that we have in our solar system and possibly in the exoplanets out there. The thought that there's life out there is a tantalizing one, and something I think that a lot of us believe in, but people like Caitlin and Micheal are scientists trying to find the way to, find evidence of life out there, and so of some sort. And I think they're going to find it. So here we go. Caitlin, I'm going to turn it over to you. You are muted, Caitlin.
Yep. Yes, I am. How about that?
I did that twice last night. So.
Oh my goodness. You would think that after many months now that we learn how to unmute, but no, right. Alas, no. But Scott, thank you so much for being my co-host through these many months now through 7 months of science. This is such an exciting program, and we're so excited to have very interesting speakers all across the solar system and then in the far reaches of galaxies and beyond. So it's my great pleasure to welcome Dr. Mike Malaska out of NASA JPL. He's been a fantastic colleague of mine for the past couple years now. Actually, we've definitely had a lot of fun with planetary science, and it's just so fascinating to watch all of his grand adventures through social media. And he's also a fellow solar system ambassador, just like you and me, Scott. So he definitely supports science communication and outreach in this case. So when he found out about 7 months of science, he was all ready to go. He was like, yes, let's do this.
That's cool.
So it's my pleasure to welcome Dr. Mike Malaska of NASA JPL. The stage is yours.
Cool. Thank you so much for the invitation. I really appreciate it. I'm going to just jump right into the presentations. Is that okay? Can I do it? Okay. So do I pull up the slides, or do y'all have to do it? I do it? Do I do it? I'm doing it. Done. Okay. Right. Very quick intro. So I grew up, I went into chemistry and I went into the pharmaceutical industry for about 20 years. And then after I saw the Huygens mission, or Cassini-Huygens land on Titan, I kind of started to go kind of gaga over Titan. I was always kind of halfway interested in planetary science. I was a member of the Raleigh Astronomy Club. Then that sort of really pulled me into becoming a solar system ambassador. I got more and more into planetary science. And then eventually when the opportunity hit, I ended up moving out to JPL as a postdoc. That's where I interacted with Caitlin, and we actually produced, co-wrote a paper describing an interesting terrain type on Titan that results from dissolution from these rains. And the other thing that I've gotten involved with is astrobiology and looking for life in the ocean worlds, particularly in the deep subsurface of some of these places. So let me dive right into that presentation, which is where we start talking about. This is where I start moving the slide. How do I move, Mr. Slide? Advance the slide. Go. Oh, poop. All right, so I'm going to have to stop sharing and see if I can regain control of my PowerPoint. Always a good thing. There we go. Share. All right, seen that. Cool. Okay, so we're going to talk about the deep ice, and this is the area before you get to the subsurface ocean. So a lot of these ocean worlds, we call them ocean worlds. They're icy moons on the outside. And hello. Wow, gosh. All right, guys, bear with me a bit. I am struggling here. For some reason, it doesn't want to advance. Technical glitches. I'm going to try to redo this again.
Yeah, maybe you don't go in presentation mode.
Let me try. I've relaunched it. Let's see what happens. Okay. Ha-ha! Okay, we got it. All right. So we've got all these.
You have to share it.
Oh, geez. All right. So let me do this. Share. Oh my. Wow, dude. What is going on?
How many tabs did you just open?
How
many dimensions did you just open?
Oh, you know what? I wonder if that was a problem. Wow. Ooh. Something ain't right. This is kind of spooky. Let's do this. Try this. I am so sorry for all of your audience out there who is watching.
They're happy that you're here.
Okay. Well, all right. We're trying this again. Okay. So getting back to around the solar system, we have all of these icy moons and worlds. And what's really interesting is if you take the amount of water and ice combined and you melt it all down and you compare it to the amount of water on Earth, a lot of these places have much more water than on Earth. So I know we like to talk about Earth as an ocean world, 70% of the surface is water. It's really just a thin little puddle. when we start comparing to some of these places like Titan and Callisto, I mean, we're talking hundreds of kilometers of ice, and then underneath that, maybe 100 kilometers of water. And then for Titan and Callisto, and we can talk about that in a second, but maybe hundreds kilometers of ice again down below the ocean because there's just so much high pressure. So there's a lot of water out there. And we know that water is really important for life as we know it. It's the main solvent that's used for moving molecules around. So it's a really good thing to think about. And when we start looking and comparing some of the different places with the oceans on Earth, we really don't find anything wrong with the concept that some of the bugs and microbes on Earth could survive just fine in some of these ocean worlds. So these ocean worlds are just absolutely great places to look for life. I'll introduce a few of them. So one of everybody's favorites is Jupiter's moon Europa. It's a beautiful, bright object in your telescope. And the reason is because it's so coated up in ice. There's kind of like this weird coloration as well, which might be from salts, which have been blasted by sunlight and may actually be fresh ocean material that kind of came up and then got exposed. We think that ocean might have some hydrothermal vents deep underneath it. And so that might be providing some nice little energy inputs. If you have a crust on the surface that's being blasted by ultraviolet radiation, you're actually going to end up making some oxygen. And if you have some type of crust moving down into the ocean, you might end up getting a lot of oxygen in the ocean. In fact, somebody said that the amount of oxygen in Europa's ocean might be the equivalent to the amount of oxygen in Earth's ocean. So that gives you a lot of the chemical fuel that you might need for life. And all that energy moving around could be really great for making neat chemical gradients. So it's a very good candidate for life. And it's also very close to Earth. So it's easy to visit. A little bit further out, we have Saturn's moon Enceladus, which is a tiny little world. I mean, it's only, it's not that big, right? It's about the size of Great Britain in terms of size. But the beauty of it is it's very tidally active. So it seems to be moving, flexing around, and then it causes it to heat up, have little fissures in the ice, and it is actively erupting jets of water, of ocean water out into space. So if you have a mission and you don't want to go drilling through a few hundred kilometers of crust to get to the ocean, Enceladus might be the best place because it's basically spewing free samples out throughout the solar system, out through Saturn's system. So you could buzz it with a spacecraft and maybe capture some of those grains and interrogate the deep ocean and see if there's any microbes coming out, spraying out in some of those grains. So it's a really good target as well. And then one of my absolute favorites, and this is what we saw in the preamble for this discussion, was Saturn's moon Titan. So it's a very large body. It's I think it's the second largest moon in the solar system. It's bigger than the planet Mercury. It has its own atmosphere, and it has an atmosphere that has nitrogen gas, a lot like Earth, but it also has about 5% methane. And that methane gets blasted by the sun. It gets converted into those really complex molecules, benzene, for example, but there might be even more complex ones as well that are being formed. And they end up going down to the surface. And so you could have all of these organic molecules on the surface. You have a very thick ice shell. Below the ice shell, you have an ocean. And we are trying to figure out through the NASA Astrobiology Institute node if there's any way that organic materials could get through the ice and get down to the ocean and make it a really great place full of organic molecules. And it turns out that we really don't need to do that, because it turns out when Titan formed way back when, it probably had a bunch of comet stuff come together and cook deep inside the core and percolate out into the ocean. And when we do the math, it turns out that there may be more organics in Titan's ocean than there are in Earth's ocean. So getting organics down into the core from the top isn't a problem. It just comes out naturally from the percolation up from below. So we have these three really fantastic ocean worlds where the ocean and the ice might be really interesting places to investigate. So one of the things that's really cool is thinking about ice itself as an actual habitat. And if we ever do have a mission that goes to these places and drills itself through the outer crust, you're going to get to a point where the ice is maybe convecting around, maybe it's a little bit warmer, and that might be an actual habitable environment. And that might be not very far below the regular surface crust, maybe only a few tens of kilometers rather than the 100 kilometers that you'd need to get down to the ocean. So that warm, deep ice, that might be the very first habitable environment that you hit on your mission going down into the deep ocean. And so that's one of the things that we want to start thinking about and exploring to see how would we, what's that environment like? What kinds of things can live there? How can we detect things that live down there? And then how would we actually pull together the technology to pull off this really awesome drill mission to look for life on some of these ocean worlds? So let's talk about the deep ice. This is the deep, deep, really high pressure, cold environment where we might actually find some of those microbes. So describing it, there's no sunlight. You have high pressure, you have low temperature. You might have very tiny little physical microenvironments, places between the ice grains where little microbes could exist. And you also have chemical concentration. So to really think about what it's like down inside the ice, we need to start thinking about our own ice and also think about the formation process of how ice actually forms. So on Earth, when you have ice that freezes, whether it's in your freezer or a cold pond or snow coming down and packing to a glacier, the story is really similar where you have ice grains that kind of come together. For ice grains that are being compressed into a glacier, they'll sinter and kind of compress up. If you're having an ocean or a lake that's freezing, you'll have ice crystals that grow downwards. That's your freezing front. And as they grow, the slower the ice grows, the bigger your crystals are. So you get these really honking huge big crystals as you get closer and closer to that magic freezing point of equilibrium, right? And you can see that in this picture here. Here's a cross-polarization microscope. The very top is like all these little tiny crystals trying to run around and find their little happy place in the crystal matrix. As things progress and the ice goes down, your water temperature drops, the crystals start getting bigger and bigger, and the crystals, water molecules have time to find their happy little perfect place to make a big honking huge crystal, and that's kind of what you see. And it's that process of making the crystals as it grows and expands. As things are nicely forming, only water ends up going to make these crystals. Anything that's not water gets excluded out of the crystal matrix. So if you have water with a little bit of salt, as things progress, you'll get beautiful pure crystals of water growing, and it squeezes all those salty materials into the little zones between the ice grains. And if you have organic molecules or stuff like that, it squeezes into them as well. So that makes as long as it doesn't freeze, right? Because you are, as you increase the salt concentration, you lower the freezing point. So you have these little tiny micro pockets between the ice grains that are liquid, very salty, and just absolutely chock full of organic food or salts for these microbes. I have an example here where they used a fancy spectrometer, and they basically shot a laser and they looked at the regular big ice grains, and then they looked at the little tiny channels between them, and they measured the chemical concentration. And they found that compared to the ice grains, those little tiny veins were concentrated up like 200 to 400,000 times higher.
Wow.
So thinking of sulfate or nitrate, I just convert that into the word pizza, right? If you're a microbe and you want pizza, right? Is it better to have like one or to have 200,000 or 400,000? And it's way better to have a lot more. And so you have these little tiny microenvironments that can exist that are just chock full of these fun, wonderful molecules. And here's a picture that's really cool. This is actually one of my favorite pictures ever. It was taken by Karen Young's group. This is sea ice up near, I think, the Chugach Sea, if I recall. They collected it in winter. And what it is you see this beautiful image on the left, big center picture, of these three grains coming together. And there's what's called a triple junction, right? Between kind of like the little pocket between all these three grains. In the upper right, I have a visible image, and you can see that little triangle pocket, little tiny thing, right? I mean, it's only like, 10, 50 microns across. This is not even the width of a human hair. They lit it up with a fluorescence stain and lit it up with a fluorescence microscope. And you can see inside that pocket, 2 little tiny bacteria basically hanging out. And somebody did the math, and they found that, 4 little tiny bacteria living at low temperatures, a little micropocket like this, chock full of all those nutrients, can survive for about 10,000 years.
Wow.
So you have these tiny little pockets that can last for a really long time. It's a happy little environment.
Wow.
Now picture how many grains there are in the Greenland ice sheet, and picture how many tiny little micropockets are there, and you get a really big idea of just how much of a landscape we now have to explore on our own planet of all of these things. Right. So there's another effect too, and it has to do with size. So as these ice grains are growing, if you're small, you can basically surf that progressing ice front and stay in that liquid zone. If you're too big, you end up getting stuck in the ice. And that magic number, and these people did it with these little polyester beads of certain sizes, turns out there's about 5 microns. So if you're a small bacteria, less than 5 microns, you're going to end up getting squeezed into that microbrine channel pocket perfectly well. If you're too big, you get stuck in the ice, and that's not a good place to be because it's also pure water ice, so there's really no food. So what that means is it's very, very, very good to be small. So bacteria in ice are going to want to be tiny. And here's a comparison of some cell sizes. So I have a red blood cell, big 8 micron thing. I have E. coli, about 1.5 microns in length. Staph A, about 1 micron. The smallest bacteria that I know of, normal, is a sexually transmitted disease, Mycoplasma genitalium. Unfortunately, we're going to be hearing a lot more about this. It also has the smallest genome. It only has about 255 genes. So that's really the minimal set of genes that you need to have a functional free-living bacteria. And then I compare it to the ones that we see in ice that are even smaller than that. They're tiny. I have a picture way up here that was isolated from the GISP-2 core. This is a big core that was taken in Greenland. And you can see these little 0.2 micron size filter holes. And 2 of the bacteria were effectively touching each other, and they didn't end up going down the hole because they were just too big. But if it was just a single bacteria, it's almost about the size of that 0.2 micron hole, and it would have gone right through. So one thing we should think about when we're thinking about life in an icy environment on Earth and in other worlds is it's going to be really small. And we have to kind of get used to thinking about these tiny little micropockets and these tiny little bacteria that live in those little micropockets. I have kind of a picture here, a rogues gallery of different bacteria that have been isolated from GISP-2. This is down about 3 kilometers deep. And I'm going to give you these special numbers. It's below freezing. It's about 264. I use the Kelvin scale just to keep it universal. So that's about -9 Celsius. These guys are alive, right? So they're not frozen. They're living in a little micro pocket, and we've taken them and melted them and shot them through a filter to isolate them. They also live under high pressure. So this number is the equivalent of about 10 to 15 kilometers deep in Europa or in Titan. So there's bacteria that live under the pressure and temperature conditions that we would expect in some of these ocean worlds. So when we're talking about whether those are habitable environments, yeah, absolutely. I will push back and say, yes, it's a habitable environment. And now the big question is, are they inhabited? And that's what I think we want to start looking for. Right. So let's look at that GISP-2 core. So here's a slice through the Greenland ice sheet. At the very top, you have snow compacting down to ice. There's an area where it's below this magic number of -20 Celsius. That's really too cold for life to survive, or to not survive, but to actually be actively metabolizing. So it can be preserved. They're in there, but they're in the deep freeze. They're not doing anything. You get way down there, about 2.5 kilometers below, and it starts to warm up as you're getting closer to the surface of the Earth. You have that geotherm, always keeping things warm. And there you do have, it's called the warm, clear ice, and there are microbes living down there. It's a very happy, habitable environment, maybe 1 e to the 6 cells per cubic centimeter. And then you get below that, and you get within like 50 meters of Earth's bottom crust, and it's really warm and toasty comparatively. There's a lot of nutrients flushing through there, and you have a huge cell population, like 1 e to the 8 cells per cubic centimeter. That's pretty close to the amount of cells in your yogurt. There's a lot in there. And it's so many that if you guys ever look at climate records for Greenland, it always goes down to a certain point, but it doesn't go much lower. You can never tell the climate from way back in time in that 3-kilometer cutoff because there's so many bacteria living down there, they throw off all the isotope ratios. That you can't do a climate reconstruction just because there's so many bacteria living down there. And it's not subtle, it's screamingly obvious. It's like, oh, 0.2, 0.3, oh my God, it's 50, right? It's a huge number from some of these values. So bacteria are actually down there doing things. Okay, so now we're going to talk about the numbers that we would expect in some of the ice. So I've got kind of a plot here, kind of trying to range the numbers of cells that we have to think about. So effectively, medically sterile is about 0.1 cells per cubic centimeter. That's sort of like an accepted standard for USP. Your laboratory work that you're doing, Caitlin, has some sort of a fancy UV filter behind sterile water, millicue water, that still has cells in it, and it's about 0.1 cells per cubic centimeter. So when we think about instrument levels for detection that we're used to for Earth, we're about 100 cells per cubic centimeter. The human body has about 2.5 to e to the 8th cells per cubic centimeter. I mean, these are big mammalian cells, so it's you can have a lot of them in there. And then if you have like little tiny bacteria, your absolute total limit is 1 e to the 15. When we're talking about the ocean worlds, we normally tend to think of about smaller numbers than on Earth. The number that we've kind of been using is about 1,000 cells per cubic centimeter, maybe in an Enceladus ocean, which is maybe 100 times less than Earth ocean or in the ice. Titan might be an exception. We're still trying to work that out. But if you have the amount of organic molecules and the energy that you might have in Earth ocean, you might be able to support a really high load of biology down there. So we're still trying to think about how many cells could be down there. But when we're thinking about this, we're trying to do it so we can scale our instruments and our technology appropriately so we can find those little tiny bacteria at a fairly low concentration. Yeah, so the other thing that we found out when we did some of the work looking at the Greenland Ice Sheet, and when I say we, I'm referring to the scientific literature at large. I didn't do this. I wish I did, because it was really cool work. And they discovered not just, a couple types of bacteria, they discovered whole different types of phyla down there. You got proteobacteria, you got archaea, you have even some fungi were found down there as well. So there's a huge biota that lives down there. And the other thing that's cool is if you notice, they were taking sections of core, 50 meters apart, and they found huge differences in diversity, even with just a little bit of 50 meters of difference, vertical, right? So not only is there life down there in the ice, but it's diverse. It matters where you go. There's micro pockets, maybe there's huge pockets. It's a whole ecosystem down there, and we know so little about it because we've only poked a few holes down in there. So it's really a right place for exploration, and it's down there, at temperatures 252 to 264 Kelvin, 25 to 30 megapascal. So these are about the same conditions that we would expect in the ice. When we think about the subsurface ocean, right, it's going to be dilute. It's going to be, stuff mixing around. It's ocean over here is going to be about the same as the ocean over here. When we're thinking about the ice on Earth, there's huge differences just 50 meters apart. So when we have, hundreds of kilometers of ice and maybe, 100-kilometer thick ice shell, the potential for all of these different very wild microenvironments is massive. So ice is really going to be one of the best places and most fun places to explore for life on these ocean worlds and on Earth as well. So here's a picture that talks about the different ice shells. If we take kind of like a vertical transect through the different worlds, I'm going to put Earth and Mars sort of on the same kind of story where you have snowfall or deposition of ice onto the surface. Enceladus, Europa, and Titan, you have ice, but then you have an ocean underneath it. Enceladus is fairly boring. You have an ice crust and it probably doesn't move around 15 to 30 kilometers. As you get to certain larger sizes, you actually get to the point where you actually have convection, almost like a glacier moves on Earth. You have actually convection moving through the ice lower, and then it hits up kind of like what's called a stagnant lid, where your upper crust is probably kind of boring and cold, but the lower section is moving around, and that tends to keep the temperature about the same. It's moving around fast enough to distribute the heat. So, once you get below that stagnant lid, like 15 or 20 kilometers in, you now start getting to where it's nice and warm and toasty enough to maybe have life going on. Same deal with Titan. You have maybe, 50 to maybe even 70 or 80, depending on the model, depending on the composition of convective warm ice moving around. So that's a lot of ice for a potential environment. Titan is a little bit weird because it's so big, it can actually have high pressure phases of ice, which are beneath the ocean as well. And it turns out that we think those could be convecting as well. It's a little bit weirder environment, and we're still trying to figure that out, but that actually might be a habitable zone as well. I don't know the rules about how much it would be similar to the more normal ice 1H that we deal with when we're talking about terrestrial life, for example. But that's something that we need to study as well. So Titan could have a lot of different cool icy environments just because of its sheer size. When we start thinking about how this all works, again, you have this crystallization. So you can imagine that at the surface, it's a big planet, right? So you have very cold ocean, very cold ice shell, everything's at equilibrium. You could have huge, massive ice crystal blades coming down, maybe meters, right? As it kind of collects and compresses the ice, you're going to have this concentration effect. You might have some chemical reactions actually happening. And eventually you might actually have convective transport. So all of these little isolated micropockets might actually be mixing and matching all over the place in Titan's ice shell. So you have these potential for these huge environments that could be changing and swapping and changing ingredients and moving around. And some people have even thought that it's possible that this might be a potential way that life could start, where you have these chemicals that are actually in a dehydrative environment, actually joining, linking together, swapping slowly, and actually causing kind of like almost like a cell-like structure, which could then eventually kick over to a formal evolution of life. So these are kind of neat ideas, but it gives us another way that life could start on an ocean world that doesn't need a hydrothermal vent, because you have this huge shell of ice that could be involved at this point. So the possibilities are huge, literally. Okay, so one thing, when we go to these, as you go down, you have more stuff on top of you. So you have to worry about pressure. So on Earth's oceans, you get to maybe about 50 megapascal at the bottom of the ocean. Europa, as you're working yourself through the ice crust, you get to about those pressures as well. Titan is bigger, and the ice shell is a lot bigger as well. And as you work your way through the ice crust, as you get to that 100 kilometers deep, you start getting into a pretty high pressure regime of about 100 megapascal. So that's 10 times higher pressure than even the deepest parts of Earth's ocean. So you might think, well, is that going to be a problem for bacteria? Some of the work that has been done previously has been looking at some of the bacteria isolated from the deep areas of Earth. There has been also some work looking at creating artificial pressure chambers and seeing just how high you can take bacteria before they freak out. And I can report that some of the work that we've done as part of our NASA Astrobiology Institute, working with some researchers at University of Illinois, Chicago, we have a pressure setup, and we have been simulating Titan ice, Titan oceans, and the bottom of Titan oceans. And our current record is taking bacteria up to about 800 megapascal and holding it there for about 15 minutes, and they survive. So, pressures may not be a big problem. So we don't need to worry so much about pressure, but I think we might need to worry a little bit about temperature. So the magic number for terrestrial microbes is about -20. So we need to kind of look at all these different temperature plots of these different worlds and see, at what point do you go below -20? At what point do you cross over as you go closer to the core of these different worlds? So I'm going to say a bad thing about Mars. I like to do that, but it turns out that there's polar caps on Mars, and Mars is really cold. And you never get to a point in those polar caps where it crosses that magic -20 Celsius threshold. It's just too cold. So you might have life preserved in the ice caps of Mars, but they're preserved. They're not alive. On Earth, it's preserved until you get a few kilometers down, and then you cross that magic threshold, and those bacteria can now survive and have a viable ecosystem. On Enceladus, Europa, and Titan, it does seem to cross that magic temperature of about 250, about maybe 10 to 5 to 15 or so, depending on the body. But you get around 20 kilometers below the surface and you're in the good. You can have a viable ecosystem living in the ice. So what it is that actually drives that magic number is you need to have cells that operate. So they need to be taking energy, doing the cellular thing, doing metabolism, and moving materials from one side of the membrane to the other. So if you have a cell which just freezes, it undergoes a glass transition, everything just kind of stops right in place. That's okay. That's cool. You can take a cell, you can freeze it down, you can thaw it back up. Biologists do this all the time by keeping cells at a -80, freezing, thawing them up when they need them, putting the culture down. It's all good. The problem is when you're right close to that transition where not everything is frozen out. So, for example, if you have active metabolism, but your membrane has frozen out and it doesn't allow things to get across the membrane, but your cell's still trying to live, that's bad. You end up killing the cell. Or what if you have a crystal that stabs itself through the membrane and ruptures it? That's bad. That kills the cell. Or what if you have crystallization that comes from the inside and goes through? That's bad. Mm-hmm. Another thing that's weird and kind of non-obvious is that as you freeze, the water likes to go into those crystals. That actually causes desiccation. It dries it out. So you can actually suck all the water out of a cell and that'll kill it. And that's bad. So it's right at that transition temperature where you're not totally frozen out on the inside, but you're still kind of trying to operate. That's very bad. So that's why having a way to control how a cell behaves and actually make the cell freeze at the right time can allow the cells to survive and control basically their own destiny. And cells do this a bunch of different ways. One of the really amazing, cool ways is that some cells can actually control ice formation. They have these proteins that latch up with the crystal structure of ice. For people who are really hardcore, it actually forms a clathrate layer around the cell first, the first initial layer at the protein interface. Then it goes to a regular ice-1H crystal structure, and that allows the cell to be able to kind of control its own destiny and decide when it's going to freeze and when it's not going to freeze. What's cool is that this has a lot of commercial value. One of the things is by snowmaking machines, for example, is one way. You can add these proteins, and that will initiate snowmaking for ski areas. You can spray this on some produce and prevent, that horrible damage freezing that can happen the same way. The other thing is, and I love this application, is by mixing in some of these ice binding proteins, you can actually control the size of ice grains that you get when you're making ice cream. And the idea is if you do it right, you can make that absolute perfect, silky, velvety ice cream that we all just absolutely love. So, this is why this research is really good, right? So there's a lot of applications for ice binding proteins, but the same problems that we have here on Earth with microbes controlling their freezing, you would think that maybe some of those cells on other worlds might have developed the same way of controlling it. So, trying to think about how cells survive in these extreme environments on Earth, and cells in other worlds are going to face the same problems, and they might come up with similar strategies, maybe not the exact same molecule, but they'll have basic same ideas of controlling where those little OHs are poking out and how the ice crystals will align to be able to protect itself from freezing. Okay, so let's talk about the life that lives down there, right? I got a picture here of Planecoccus halocryophyllus. These guys live at like minus 18 degrees Celsius. So these survive just fine below freezing. They live in those little microbrine pockets. They're good. This particular bacteria was isolated from GISP too. So yeah, you can have life below freezing. Totally good. You can have weird metabolisms. So here's a bacteria. It's one of my favorites, Shionella putrescens, isolated from deep below the Earth, and it actually eats metals. It can take iron and convert it over to iron 2. It can take uranium and convert it over to a different oxidation state of uranium. It is thought that these bacteria, or some like it, may have been taking some of the soluble uranium flowing around groundwater in the southeast, eaten it, and then pooped it out as the insoluble uranium. And that's where we got all of our uranium deposits from. It's basically Shionella poop. So these bacteria might be very economically important. We just didn't know it. So these guys are multi-fuel bacteria. They can take any metal and convert it from almost any oxidation state to another oxidation state and get their energy out of it. So bacteria like this could do chemical metabolism on an ocean world. Another one that's one of my favorites, actually many people's favorites, is a bacteria that can take acetylene, add water to it, and it makes acetaldehyde. Acetaldehyde is the smell that you have after you drunk alcohol. You convert some of the ethanol over to acetaldehyde. These bacteria are taking acetylene, hydrolyzing it with water, making acetaldehyde. That's a downhill reaction. That's how they make their living. There is a bacteria called Pelobacter acetylenicus. It can eat acetylene and use it as its own energy source and its sole carbon source. So this bacteria, you could probably put it down in the deep Titan Ocean and it would be totally happy. I forgot to mention acetylene is one of the major products of atmospheric photolysis on Titan. There's huge amounts of acetylene there. So these bacteria would be very, very, very happy down there. We think as well that the enzyme that does this, genetically, it looks like it's ancient. So this could have been one of the precursors or one of the first forms of life that started on Earth when it was still running off of chemistry before it kicked over to using photosynthesis. So this could be a very ancient way of life starting, and it could have happened on Titan too. So this is all good, right? We have another bacteria. Here's one of my favorites. That's actually my hand. I was in a cave in southern Mexico, and we had all these bacteria where we had hydrogen sulfide, oxygenated water, and where they mixed, we had these flocculent bacteria rubbing up against your legs. You pull it up. We called them phlegm balls because that's kind of what they look like. Really gross, little slimy guys. They take rotten egg gas and oxygen, and they pee out sulfuric acid. And that's a downhill reaction, and that's how they make their living. Okay, so you take these guys, and now picture this. This is a picture of that cave. So here's a bunch of us standing in a sulfide spring, and you can actually see the difference in color between the sulfide spring on the left and the oxygen-rich spring on the right. Just downstream from where they mixed, literally a stone's throw, was where that bacteria was plucked out. Picture Europa's ocean. You have the hydrothermal vents on one side spitting out H2S. You have oxygen coming in from the outer crust as it's being subducted from UV photolysis. So you have oxygen, you have H2S, they're mixing. You have the same environment where these little bacteria can make their living. So you could take that bacteria, probably find some place in Europa's ocean where it would be very happy. So yeah, there's lots of different metabolisms, and all of them could be totally valid on the ocean worlds. Other wild stuff, here's a hydrothermal vent. We did not know about hydrothermal vents when we sent the Viking mission out to Mars. So a lot of the stuff, hydrothermal vents, life in the clouds, life in ice, all of this is very, very recent work and understanding of learning how biology on our own planet works. There's a huge chemical gradient as these hot fluids come spewing out of the deep earth, and you have all of these bacteria, tube worms, and things living off of that energy gradient. If you have hydrothermal vents on another world, you could have the same type of energy kicking in, and you could have the same type of biota that might be able to survive there as well. And one of the more fascinating and amazing discoveries is hydrothermal vent bacteria off, I believe this was off the Oregon or Washington coast, where they found these bacteria that are doing photosynthesis off the glow of the hydrothermal vent in infrared. So you can have plants that don't need sunlight. So you have these ocean worlds that have hydrothermal vents. You can still have an effective photosynthetic process. It just doesn't use regular light wavelengths. It uses infrared. Totally cool. So these are the kind of things that make us need to rethink how we think about biology and where biology could survive and exist on other worlds. Okay, so here's going down deeper. We have these bacteria. This is a Formicutous bacteria related to hydrothermal vent bacteria. These live way down deep. They're harvesting some of the hydrogen coming from radioactive decay. Genetically, these have been cut off for surface life for about 15 million years. So below our feet, way down there, there is a whole nother biosphere that we're just only starting to learn about now. So anything that lives way down there deep, that could be maybe if it could survive the temperature and pressure conditions of Europa or Titan, it might be okay. If it can survive even higher pressures, it might be something that might be way down deeper in the core where we're not even talking about it yet. But it's showing you the frontiers of life and of astrobiology. We don't know how deep the biosphere actually goes. A lot of people have said maybe 10 kilometers because pore pressure squeezes it off, but we really honestly don't know. And it's going to take more and more difficult exploration to figure that out. But the implications of it are huge, because now you could think about places like Mars, maybe even Venus, maybe even, Mercury or some of these weird rogue planets. As long as you have heat residual in the core and you're down deep enough, it might be totally protected, right? We talk about, sterilizing the surface of the Earth. These guys have been down there for 3 to 15 million years. They wouldn't know, right? They're protected. You could wipe out all surface life and these guys would be down there just fine. So you cannot easily kill off life on Earth while these guys are still alive. So all sorts of really neat things to think about as we go exploring. Probably one of the more fascinating discoveries in 2010 was from these brine pools that they've discovered down in Gulf of Mexico. There's also some in the Mediterranean. It's kind of like a hydrothermal vent, but it's more cold fluids that are percolating up out of the earth. There's just chock full of methane, chock full of hydrogen sulfide, a lot of stuff in them, super saline. They're so saline that they actually form a different layer than the regular ocean water. So it's almost like the Talking Heads song where, you go down to the bottom of the ocean and there's water at the bottom of the ocean. There's these lakes down there. And as the ROV came down and poked at the surface, you could see waves rippling across these lakes, even though you're like 2 kilometers below the surface. And all of those things you see on the edge, those are mussels that are all living off of the bacteria that are eating the hydrogen sulfide, that are eating the methane. And one of the most amazing things that was discovered in 2010 is this little critter over here on the right called Lonicifera. It's kind of its own phylum, but this is an anaerobic creature, but it's multicellular. We used to think that, things that are living without oxygen have to be very simple, have to be very, primitive. And know that you can have something that's actually multicellular, but lives completely without oxygen, like this guy. So when we start thinking about ocean worlds of other places or alien worlds, you don't have to have just simple microbes. You could actually have these complex multicellular creatures that actually exist. Whew, so a lot of stuff has changed in the last few years with learning about as we probe the extremes of biology. So now let's talk about deep ice habitability. Is it a habitable world? So I have a couple plots here. Effectively, what it shows is temperature and pressure. There are bacteria on Earth that live under those conditions. And I can name names, and here are some of them, but they all live under these very low temperature and very high pressure conditions. So you could probably put any one of these bacteria in the corresponding location on Europa, Titan, or even Enceladus, and it wouldn't know the difference as long as you give it the right little tiny micronutrients. So we can now say that it's probably a habitable environment. And then the big question now is it inhabited? And now the big question is, how do we go figure that out? So we can start picturing now a huge astrobiological investigation showing, my favorite place, Titan, where you have these organics coming down. Maybe they work themselves through the ice shell, maybe not. You have a bunch of organics down there in the ocean. You have a biota. Maybe they are coming up and putting in little ice lenses. And all you need to do is take your drill mission, land on Titan or any of the other ocean worlds, drill through, and then look for life. So the question is, how do you actually pull that off, right? We have a mission that's going out to Europa Clipper. It's called Europa Clipper. It's going out to Europa in about the 2020s, launching later this decade. The next follow-on mission is a proposed mission concept called Europa Lander. But then the next natural mission beyond that is going to be the deep drill. And that's where you're going to be going all the way through the crust and trying to look for life on your way to the deep ocean. So how do you look for life? What are the signals and things that you would want to look for? And the thing that's probably the easiest is to look for chemical signatures. And I'm not going to get too boring on you, but one of the things that we do is we have a pretty good idea about how some of the chemistry might work on some of these ocean worlds. And what we want to do is look for anything that's a little bit different that might show that there's life there that's changing things. So I like to think of a very simple definition of life is that you have food and light, you convert it into chemical energy, you take that chemical energy, and then you make structures, biomolecules, things that are useful for you, more bodies. So when you make those, you're going to make things that look different. So for example, I have a diatom here that we isolated from the Greenland Ice Sheet. You see that under a scanning electron micrograph, you're going to go like, oh yeah, that's weird. There is no way you could make that naturally. That is probably something directed it and made that particular shape function thing. Maybe a little bit less obvious are some of the chemical signals that you might see. A lot of these are compounds, amino acids, carotenoids. They're fairly complicated structures. And you would say that, you know what, I'm probably not going to make a lot of that material just by itself. So if something's making those, and I see an abundance of all of those, that's probably some type of a life process that's happening. And it could be either something that's useful as a body part, or it could be maybe something that, I ate this and I now threw away the pizza crust or whatever, and that's what I'm seeing. So basically running around looking for pizza boxes or pizza crusts or even pizzas that are halfway eaten can give you a clue that there's something down there that's alive. It may be different from Earth life, so you don't want to end up looking for the exact molecules, but you might have the same basic strategy. So we're going to look for maybe membrane forming molecules or molecules that might be able to link up and make proteins. Not the same amino acids we use, but different ones. So when we do that, we're going to be thinking about, what gets made naturally, what has an energy barrier, and then one of the hallmarks of life is it lowers the energy barrier and it makes different molecules than you'd normally expect. So it's going to be looking for those different molecules. The most hardcore slide chemical-wise is here, and we have these things called amino acids, and they come in two forms. They have a handedness to them, and if you have a natural or a non-biological process making them, you're going to make both forms, a left hand and a right hand form of these two amino acids. If you have biology, it's really only going to want to use one form of them. So it's going to really lower the energy barriers to synthesizing that to make it in abundance. And so you would expect to see a throw. You would expect to see more of one form of amino acid than the other. So we can go look for that with some type of a detector and see if you have a different type of molecular pattern or a different type of chirality in this place. So one of the things that we've presented is what's called the abiotic synthesis pattern. So you use things like fatty acids for making membranes. And if you make these without biology, you get, it's harder to make really long ones. For biology, it likes to have the long ones of a certain length to fit your cell membrane structure. And the way that it builds it for plants in particular is it always adds them on in groups of 2. So if we see this weird even or an odd pattern, we know that it's doing something funky that has to do with the biosynthesis, and that gives us a clue. So all of these are kind of giving you kind of the idea of what sorts of patterns we would look for to be able to detect, to say that, you know what, there's probably a good chance that what we're seeing down there is life or is the result of a life process. Now, the other part is getting down there in the first place. And this is where we start talking about the technology that can drill you through the ice crust and get you down to get those instruments down where they need to be able to detect life in the first place. So I've been part of a program that's basically trying to build one of these huge stacks. We call it almost like an instrument train. So here's one idea of being able to stack a bunch of these different subunits together to make something that could drill through the ice and then get down into the ocean. And one thing is we have this drill on the end that's maybe the slush drill or somebody else's made by Honeybee Robotics. We have an instrument that we've been developing that would be useful for trying to detect things actually in the ice to say, oh, here's something interesting. Then you have something that grabs the sample out of the ice. Then you have another thing that does the really hardcore chemical analysis that says that, yep, here's our pattern for life. And then you might have some sort of a fancy robot that once you get down to the ocean can then go swim around and do some serious long-term mobility. So the thing I'm going to talk about for the next few slides is talking about how we develop this initial detection system, which is the front end of the instrument train. So we decided to use a UV spectrometer because UV is super, super, super sensitive. We can see things at a very, very low amount, like you'd expect in one of these nutrient depleted, very low bioabundance icy worlds. So we have this system that basically shoots a laser out into the ice after it follows along behind the drill, shoots the laser out in the ice, and then it detects where these microbes might be. So basic idea of fluorescence is you excite a molecule, it kicks it up to a higher energy state, it kind of drops down a little bit, and then when it drops all the way back down to the ground state, it emits a longer wavelength photon than when you put into it. And y'all can see this by shining a black light on different alcoholic drinks. I have tonic water, which just by itself it glows. So that's a really fun thing to do as a demo for kids. Energy drinks, like anything containing vitamin B12, glows green. The French liqueur chartreuse, which is normally green, as you can see in the upper corner there, when you light it up with black light, it turns red. So it's basically from a lot of these long chain phytochemicals. So we're going to basically be shooting a laser out into the ice and seeing colors glowing back at us from different things that are fluorescent. And things would be amino acids like phenylalanine, tryptophan. Here's a signature of a spectra from a microbe. And so by looking at the spectrum, we might be able to guess at what kind of a molecule it is. So here's where we went. We went to Summit Station, which is way up on the very top of the Greenland Ice Sheet. This is a picture that was taken, I believe, about 11 o'clock at night. So you can see the sun still up there in the sky. It's 24-hour daylight. It's an absolutely beautiful place. If y'all ever get a chance to go out there, I mean, it's a purely scientific research station, but I thought it was going to be just boring ice, you know, but the sky and the ice had all these really cool lighting effects. It was always changing. It was just absolutely gorgeous. Here's a picture of where we did our drilling. We were actually 7 kilometers away from Summit Station. So we were basically an hour, well, no, 30-minute snowmobile ride out from Summit Station and then a 3-hour flight to get anywhere. So there's an ice runway out there. This is our drill camp. We have a drill head that you can see poking out of the tent, and we did all of our drilling down to about 105-meter depth inside that tent. And then we had our instrument so that we could scan inside the borehole. And here's some of the stuff we saw. So here's a picture of what it looks like inside the tent. We had a little snow pit where we could grab some cores and take them out. There's our instrument, a big silver tube, and the little window was right there, kind of facing us. And what we do is we send it down into the ice, and then we shoot the laser out into the side of the ice, and we can make a scanning map and see all of these neat little weird fluorescent hotspots that are embedded in the ice. Trying to remember, I believe these were in place right about the time that Abraham Lincoln was president. So a long time ago, very well preserved. We see all of these little spots. I've zoomed into one of them, and it's pretty compact, small little thing, about maybe 1 millimeter across. We look at the spectra, and they all look pretty much the same. So it's a very uniform little blob across it. And what's neat, and it's unsurprising, is that there were all these little individual spots, and they were all spectrally unique, but they were all different too. So even at the scale of just a few millimeters to centimeters in the ice, we're seeing all sorts of little different chemical microenvironments, maybe different bugs, maybe different chemistries, but it's all different. It is not just boring uniform ice. So we're learning a lot that how varied these different ice environments might be, in addition to testing out our instrument. Here's a picture of some of the different spectral things that we saw. We calculated 15 different things. I think now we're up to about 25. Here's a picture of us raising the instrument out of the borehole for the last time. That hole goes down 100 meters, 110 meters now. And this is basically how we did it. This is us raising it up. As the instrument rotates, you'll actually see the scanning mirror give you kind of like a neat little flash. Boom, there we are. So there's the instrument and the mirror where the laser shoots out. So here's sort of our results, a map of what we saw down at 93.8 meter depth. The big lesson is that we didn't see layers like we expected. We saw all these little concentrated hotspots, zones of organics and microbes. There's this general theme when we look for life, is that life likes to clump. We don't know why, but it's just sort of a thing. And you can even see it in human cities, right? We're not spread out all across America. We're in villages, we're in cities, we're in little houses, and that seems to fit life in ice. It's in little houses, micropockets, it's in little clumps, and even larger accumulations itself. You can see that right here. It's not uniformly distributed all the way through. There's little spots here, and then these big empty spots. So when we go looking for life on other worlds, we want to be able to hit those, to identify them, detect them, scoop them out, grab them, and know that there's going to be some void spots, and know that there's going to be some really concentrated hotspots that may be different from each other at a very small scale. So the paradigm before we did this work was that, oh yeah, we'll go through the ice and we'll just melt it all, and we'll try to suck up a sample from time to time. You do that, you're going to mix everything together, and you're going to dilute it out big time. Here we see big concentration spots. Let's grab each one of those spots and see what's in there. What's in the blue stuff? What's in the yellow stuff? What's in the green stuff? I don't know, but it's all different. So it's all fascinating to think about. Okay, so I'm going to wrap it up real close to say that, deep ice, we know it's an active habitat on Earth. It is relatively unexplored. We know now that it's an analog to these other deep icy crusts, such as on Europa, Enceladus, Titan, Ganymede, Callisto, Ceres, maybe. It's going to be the first habitable environment that we're going to hit on our way down to the deep subsurface ocean. So it makes sense to look for life in that environment. And the beauty is that we can test the tech for exploring for deep ice on other worlds. We can do that right here on Earth. And in the process, we're going to explore so much and learn so much more about our very own planet, which is really what the goal is, after all, is, going to the outer solar system, understanding life out there, understanding life here, and learning about all the different ways that life can start and survive. And so with that, I'll leave you with this picture of us taking some ice cores off the Greenland Ice Sheet near Kangerlussuaq. And I am more than happy to answer any questions y'all might have. Cool.
Oh, yeah, really amazing. Really amazing. I think that you have,
fantastic.
go ahead, Caitlin.
Oh no, I was saying fantastic. We do have a couple questions here and there through all of our different portals that people can watch on, such as Facebook and YouTube, and certainly on Explore Scientific.
Awesome. So I'm more than happy to answer anything, and I don't know about y'all's time, but I'll stay into the last questions asked, basically. So we're all good.
Fantastic. I'll go ahead and start off then real quick. So I'll combine like two questions here. How might have we figured out how thick the oceans are at like Callisto and Titan and so on? And also, how do we know about the salinity about these oceans?
So those are absolutely excellent questions. For knowing salinity and depth, there's been a lot of experiments looking at the gravity field of spacecraft flybys. So you can tell, looking at some values, you probably know more about this than I do, like the J2 and K2 and all those geophysical constants. So you can figure out that there's like different layers and what the densities are of those different layers and kind of come up with some different approximate thicknesses of them. You also have, for some of these worlds, you have induced magnetic fields. So you can kind of come up with some rough ideas of salinity. One of the things that gets a little bit more complicated for where you don't have really good information is you end up with coming up with a couple of different guesses and different models. So for Titan, I'm using a lot of models that have been made by Steve Vance, and he's come up with a whole bunch of them. So we have different compositions of salts, different densities. And then when you have the different densities and the different salts, that can actually change your freezing point. So now that changes your freezing point. So then that can tell you what your concentrations are, how thick your ice shelf. And there's like a whole bunch of I guess I want to say degenerate solutions. There's a lot of possibilities. And until we do a mission such as, say, Dragonfly, where you get an idea of maybe how deep the ocean is, that's when you might be able to then figure out what kinds of salts you might see. And I'll even make it more complicated. So for Titan, you also have the possibility of salts, but also ammonia. Ammonia decreases the density, massively lowers the freezing point. Salts increase the density and also change the freezing point a little bit, but not to the extent that ammonia does. So it's like, well, how much ammonia is in there? How much salt is in there? What's the density? And you have these two competing things, and it's really hard. You just have a whole bunch of a range of possibilities. So they're good guesses at this point. How's that?
Ammonia gets very, very entertaining in models. I can definitely attest to that. So, but you're right. It's so fascinating how much we don't know, and you and I are both in the crosses between modeling and experimental, and fieldwork. We want to do more experiments and we want to do more field work to better understand the models and, if anything, improve the models.
I think like the magic word is like constrain. It's like you're not actually finding the answer. You're saying like, well, it could be somewhere in this box. So let's evaluate those different possibilities and see how it shakes out. So.
True. If anything, we're still needing to find the box. Yes. Another question. I don't remember who asked it, Scott, but someone asked, someone was wondering why these icy moons don't just sublimate away.
Because it's cold. It's really cold out there. So the surface of these places, well, they have gravity, for one thing, that helps too. But they also have very low temperatures. So maybe about 100 Kelvin or so. So you're at 100 Kelvin, your vapor pressure isn't very high. But, and I will say, when we go to like these really boring places like Callisto, where not much has happened in a few billion years, you do see evidence of sublimation and redeposition has occurred over, a long time ago. It's been really slow, but it has happened. So it's not, a totally whacked out idea, but the gravity was enough to keep those molecules from basically flying off into space. It made them be able to do a little hop around the surface to get from a warm spot to a relatively cold spot. So it's sort of like a combination of low temperature and gravity for some of these places.
That question was from Norm Hughes. Another question from Martin Eastburn. How many forms of ice is there known in our solar system?
Okay, that is a great question. So as part of our NAI, we've been working with a person named Baptiste Journeau, who is currently, he was at University of Washington, then he's at Nantes, and I think he's now in Washington now, I think. But he has been really reworking the phase diagram of water. And there's a lot of different phases. A lot of them happen at very different temperatures, and a lot of them happen at very high pressures. And I think you can go on Wikipedia and just Google up, or you can Google up a phase diagram of water, and you'll get kind of like a plot that shows all of these different zones of temperature and pressure, and will tell you what ice phase you might have. So for Titan, which is probably my favorite world, you have Ice 1H, which is the one that we're used to. That goes down to maybe 100 kilometers down. Then as you go to higher pressures, you can get higher phases. And I believe the ones that are more likely, I think, are like Ice 5 and Ice 6, and maybe even Ice 4. I don't remember. But I believe it's not super clean that it's one or the other. It might be like a mixture of several phases. I think Steve Vance has talked about what was called like the Dagwood sandwich model for Ganymede, where there could be lots of different layers of different phases, and you may even have ice freezing out in one phase in a different place and actually snowing upwards. Some really weird bizarre effects due to the density and the crystal structure of some of these ice phases. And the other thing I'll throw out there as well is that you have just pure ice that can do these things, but there's also the possibility where you have certain gases under certain pressures and temperatures. You can have things like clathrates, where you have a little cage molecule of water. And if there's something inside that cage, it can make a little bit more stable structure. So gas molecules like methane or ethane, small, cute little things like that, can actually sit inside the cage. And that's important for us here on Earth because you can have natural gas deposits that are actually encapsulated in the ice, and getting some of these clathrates, you might be able to extract them out. Or if the clathrate forms where you don't want it to, you can sometimes have problems like this may have actually been what caused the BP disaster in the Gulf, is you had clathrate forming around one of the valves, and then it basically clogged up the valve and you had a problem. So.
Oh, wow. Norm Hughes has another question. He says, is the science strong enough to get NASA to fund these types of expeditions to core drill a faraway moon, or will it be pushed off as a fantasy for years to come?
So I would love to see it sooner rather than later. I will say that NASA is funding particular drill technologies. So they've went through a formal proposal that was called IC2. No, it was IC2. It was Coltech, I think, where they were actually looking at technologies that were favoring using advanced, I might have missed the exact call name, but there was a specific call looking for doing autonomous deep drilling for a period of years. So it's exactly the kind of thing you would want to do for doing ocean world drilling operation. I will say that drilling is not trivial. I mean, even regular drilling is pretty hard, and ice drilling on a remote body, it's hard, right? But we do have missions that are going out and doing planetary drilling. So the deepest one that you're going to have is going to be the ExoMars mission. It's going to go down a few meters into Mars's crust. And you also have, I believe, the Viper mission as well, going to the moon, is going to do some regolith drilling. And then in terms of a future drilling mission, I don't know when that's going to happen, but I do know that there's a lot of people which are heavily invested in the technology to do that. So our Honeybee Robotics, we worked with them as our partner, and they are actively working on doing planetary deep drilling, both in terms of ice and also in terms of rock. So I'm hoping it happens soon.
So exciting. There's always work to be done. It's just a matter, gotta wait for that call.
Yes.
So yeah, that's how it goes.
After listening to your talk, it makes me wonder how life might have arrived. There's models of how it arrived here on Earth. Are there models of asteroid hits or something where life spread out throughout the solar system and it collected on planets? Or are there other models that where life had spawned almost spontaneously, on these other worlds?
And that's a great question. So one of the ideas, I think the term is panspermia, where you basically had maybe one place and life kind of spread all over. Mars is a great place to think about life starting, because early on in the solar system's history, it was kind of out there and it was small and puny. So it probably cooled off a lot faster than Earth did. So life could have started on Mars. And studies have shown that you can transfer materials from Mars to Earth and they'll remain pretty much intact. So it's possible that life could have transferred from Mars to Earth. When we're thinking about the ocean worlds, it's that problem of not having an atmosphere. Oh. So for places like Europa or Enceladus that don't have an atmosphere, if there was life that was riding along on a rock, blasting around in the solar system, and it happened to hit those icy surfaces, it would not slow down. And it would basically hit the ice with such speed that it would just blast itself. The kinetic energy would just fry everything. So there could have been life hitching a ride. And when it hit, it would have been blown to smithereens and that's it, game over. Titan presents a different case because it does have a thick atmosphere. And I think somebody actually did do the calculation of how many meteorites from Earth could have ended up transferring over to Titan. And it was not an infinitely small possibility. It could have happened. The only problem though, is that anything landing on the surface of Titan would be still sitting there and it wouldn't get down into the deep ocean because you have that, 10-kilometer thick crust that you'd have to get through. Now, we just had a paper that came out that talked about a really big impact on Titan, probably from a comet, that ended up actually punching itself a hole all the way through into the deep ocean and splooting some of that deep ocean out onto the surface. But this was, a rare impact, and it was made about a 400, kilometer wide crater. So it's not, your typical impact, and you'd have to have, that thing would have had to have either life riding along on it or happen to have dredged life through the crust to be able to seed it effectively. So it's kind of hard to see. So the idea is that maybe that some of the ocean worlds may have been an independent genesis of life and may have a totally different biochemistry than what we're used to. Where if we go to Mars, there's actually a chance that we might go there and it'd be like, hey, it's just like us. Maybe it started out the same way. So don't know.
Interesting. Okay. I don't think there are any other questions here. I know that people found your talk really fascinating, and it was awesome to have you on this program. So
well, cool, thanks. And if there are any other questions, feel free to hit me up on Twitter. It's Mike_Malaska, so you can bug me there, and I'll try to respond. So thank you so much for the opportunity to come out and chat. I love ocean worlds and I love to talk about it too. So thank you.
We thank you so much for chatting with us here this evening. This has been such an exciting honor to have you here tonight. I'm sure everyone is still so starry-eyed over ocean worlds and even just the definition of ocean worlds. There's worlds with oceans. Oh my goodness. How mind-blowing. And that we can study certain ocean worlds here on our own planet. Yeah. With analogs in Greenland of all places. Interesting. But thank you so much again, Mike, for joining us this evening. Thank you again, Scott, for being our powerhouse with Explore Scientific.
Yeah, and thank you, Caitlin, for bringing on all these fascinating people. So, being exposed to these ideas and stuff is really makes the mind go to work. And I think that one of the things that also is compelling to me is that in the search for life elsewhere in the solar system, we found more life here. We made more discoveries right here. Okay, so I think that we're going to find that the whole business of life is much more amazing and much more compelling than we ever dreamed. So that is just really cool. So thank you.
All right. Thank you all very much. And thank you, Scott. Thank you, Caitlin, again. And I guess I'll sign off and see you all later. Bye.
All right. So, Caitlin, do you already know what our next speaker will be at this time?
Ooh, yes, I already have October, November, bwahaha. But I'm not going to spoil.
You're not going to spill the beans,
no,
okay?
No.
All right, well, that's all right. That's cool.
It's going to be fun. I promise that. It's going to be fun.
It has been an amazing ride so far. So thank you very much.
But it's amazing how much 7 months have gone by. We have 2 episodes left, everybody. We have October and November. And then that is it for 7 months.
I thought it was 70 months of science is what it was.
Oh, my heart.
We were just dropping off a 0. So
my blood pressure just starts to go away. Yep. But you know what? Who knows? Who knows what's gonna come out in the future? Maybe we'll do a spin-off series. Who knows?
That would be great. It would be awesome.
Who knows?
Yeah,
but I need a winter break.
No problem. No problem. Well, great. So I just want to remind everybody that this weekend we have the 45-degree Global Star Party, and this is with the Royal Astronomical Society up in Montreal, the Montreal Center. They're at 45 degrees north. And then the Royal Astronomical Society down in New Zealand. So they are going to give a simultaneous Global Star Party. It's Saturday, starts at 7 p.m. Central, 8 Eastern, 1 p.m. New Zealand time the next day. So it's going to be awesome, and it's all about the Royal Astronomical Society. So what a great group. Thank you very much, everyone. You guys have a great night. Keep looking up, and we will see you tomorrow. Take care.